Hi everyone, so we're going to continue our muscular system conversation. A couple more vocab words here, but we're starting to get into the different steps that we need to have a muscle contraction happen. So one of those most important steps here is going to be this neuromuscular junction. So this neuromuscular junction, this is where a site where the nerve fiber is going to meet a muscle fiber, and which is going to help control that muscle fiber and tell it what to do. So here we're going to see our motor end plate. This motor end plate is the site where this nerve fiber is going to contain a lot of mitochondria, and the sarcolemma is going to be extensively folded. Remember that this sarcolemma is the membrane on muscle fibers. So we're going to see this motor end plate kind of be connected or very close to this sarcolemma, which is our muscle fiber here. We're also going to see a synaptic cleft. So this synaptic cleft is this tiny gap between this motor nerve fiber and the muscle fiber. So that little gap is going to be where that connection is made between the nerve fiber and our muscle fiber. And then finally, our last term here is a neurotransmitter. So this is a chemical that is going to be stored in different synaptic vesicles that is going to trigger some action when they are pushed to the muscle cell, leaving from that synaptic cleft. So if we take a look here, this is our nerve cell connecting to our muscle cell here. We'll see that this right here, this is our motor end plate. These little gaps between our muscle cell and our sarcolemma, which is, again, that membrane of our muscle fiber, that is called our synaptic cleft. And then these little blue dots that you see here, those are the neurotransmitters that's signaling to our muscle cell from our nerve that something needs to occur here. So we can see that there are many connections here. We have many different branches and motor end plates that are connecting to our muscle cell, but only one muscle cell. So one muscle cell could have multiple nerve connections here. So again, if we take a look, we can see that here's our muscle fiber. Our neuromuscular junction is here, and this is our nerve coming to meet it to give those signals here. So motor units. This is going to be these nerve fibers that are densely branched like we saw in our image before. So we have many branches coming off that one nerve fiber. So again, like I said, one motor nerve fiber can actually be connected to many muscle fibers. So one nerve can be controlling many different muscle fibers and telling it what to do. So this motor neuron and the muscle fiber that it controls is what we're going to call this motor unit. So however many muscle fibers it's connected to, and this neuron is going to be our motor unit. So if we take a look, we have our nerve here in yellow, and we have many, 1, 2, 3, 4, many different muscle fibers here. We can see at each end where we're connecting to our muscle cell, that's where we're going to see our neuromuscular junction. But this entire thing right here, this connection with our nerve cell and these many muscle cells, that is our motor unit. So that's one motor unit. And here, this connection again with many muscle cells, that's a second motor unit. So one nerve cell to as many muscle cells as there are there. So let's talk about starting these different steps and things that we're going to need for a muscle contraction. So first thing that we want to talk about is the sliding filament theory. So we learned the structure of this myosin and this actin. So what we see with our sliding filament theory is that the head of our myosin is going to form what we call a cross bridge that is going to attach to our actin molecule. When it attaches, we're going to see that it's going to bend and it's going to start pulling at this acting fiber, causing it to move, hence the sliding filament theory. So our filaments are going to be moving here. When the head is released, it can combine to another site further down and pull again. So that's how we're going to see that muscle contraction happening. And it's going to be really important to take a look at these different videos that I'm going to send out and look with me as well as we're going through these different steps to see how this contraction happens. But there are actually 2 requirements necessary for this contraction to happen anyways, which is going to be calcium and ATP. 2 really important. If we don't have one of those things, you will not be able to contract muscles. So if we take a look, we can see nothing is connected here. We see our A band, which is our dark band with our myosin and our actin molecules, our Z lines, which is defining how long our sarcomere is. So this is relaxed. We can see when we're contracting, those myosin heads are attaching to these actin filaments right here and pulling our sarcomere in. So we can see there's movement within our sarcomere here. And again, when we're fully contracted, we can see that our sarcomere has shortened a lot. Our muscle has fully contracted. Those myosin have closed this H zone that we talked about. That is just myosin completely, and we have a full contraction here. So it's going to be important for our sliding filament theory and how we contract our muscles. So since we have a contraction, we're going to talk about some of the stimulus we need from contractions. So with our neuromuscular junction, we get that signal that tells us, okay, we're going to contract our muscles now. So some of these chemicals that we're going to see, the first one is called acetylcholine. So this is going to stimulate, that's going to be one of those neurotransmitters that's being sent to tell us that we're going to contract muscles now. So that's going to stimulate our contraction. And then acetylcholinesterase is going to be the enzyme that promotes muscle relaxation. So we're going to pump in a lot of acetylcholine when we want our muscles to contract, and when we want them to relax, we're going to send in acetylcholinesterase, which is going to bind to our acetylcholine and not make it, and it will tell our body to stop contracting that muscle. So those are 2 important terms for stimulus for contraction. Energy for a contraction is going to be with ATP. So ATP is probably something you've heard before, adenosine triphosphate, so there's 3 of them. When a muscle fiber is active, ATP must regenerate. So we'll see with our muscle contractions, when we have ATP connect to those myosin heads, it's going to be converted to ADP plus a phosphate. We're going to use that energy that bond was broken with to move that head, but afterwards we need to regenerate that ATP. So what we're going to have to do is take this ADP and that phosphate and remake this bond to make ATP. So creatine phosphate is something that's going to be able to do that for us, but this is only temporary energy, so we need a more prolonged source of ATP to be able to hold muscle contractions. So if we take a look here, this is just a cycle of our ATP. So what's going to happen, we get energy from our cellular respiration. That's going to make our ATP molecules. We're going to use that ATP for a muscle contraction. So when we use that, so when we have ATP is low, we're going to see this creatine is going to be recycled here. So this ATP is coming down. This creatine phosphate is going to help us recreate that ATP and kind of cycle this, but this is only temporary. So this energy here that we're going to see with our creatine phosphate is going to be temporary. So we really want to get all of that energy and that ATP from cellular respiration. And we'll talk about that a little bit more next time. But this is where we're going to stop for now. If you have any questions on what we've talked about so far, this sliding filament theory, please make sure we reach out and ask.